Good evening, everybody. It's my pleasure to introduce our next speaker for the evening, Alexander Ivanov. Dr. Ivanov is an associate professor in the Department of Chemistry and Chemical Biology and a faculty fellow at the Barnett Institute of Chemical and Biological Analysis here at Northeastern University. His research focuses on ultra-high sensitivity proteomic profiling of limited samples, the characterization of proteoforms, protein complexes, and protein interactions, as well as the exploration of extracellular vesicles for diagnostics and therapeutic development. Please join me in welcoming Dr. Ivanov.
Oh, great. Thank you for your kind introduction. Thank you for sticking around. And Nikolai, thanks for your invitation. So as you may have noticed from the program, so I have the longest title in the whole conference, and I suggest that Nikolai introduces prizes for shortest and longest titles of the conference. And since I'll be introducing hyphenated technologies, so I use 5 hyphens in this title, as you may see. So this is difficult to beat. So definitely worth the price. Okay, so I've been told that I need to declare a conflict of interest. I spun out a company or a small startup to be eligible for STTR SBIR grants. We're applying now, and hopefully we'll explore commercialization potential for our technologies in the future. So I'll be talking about 2 directions. And Michal is here. Michal is the first one. So 2 directions in our technology development on porous layer open tubular columns. And yesterday this term came up during the discussion after Ryan Kelly's talk. So here I'll be talking mostly about porous layer open tubular columns. And either we are using them in pressure-driven technologies for separation, or in electric field-driven separations, capillary electrophoresis. And I need to mention Michal, Yunfan, and Liz. And in the previous years, Soma contributed to data analysis, and Liz, Michal, Yunfan, they generated the data. So we discussed all these possible needs for single-cell analysis and subcellular characterization, possible neonatal samples, small animals, animal samples, and so on. We discussed that proteomic characterization is more important or maybe more informative than transcriptomic characterization in many cases. And definitely there are challenges in characterizing proteins, many proteoforms, and many peptides and proteins in small samples. So, and as Alexei mentioned yesterday, in the analysis of small sample amounts, single-cell proteomics, there are multiple challenges. So at each and every step, and each and every step had to be brought to a perfection or improved, and any improvement would matter. In this talk, short talk, I'll be only discussing our improvements in separations, either LC or CE. So the point I'm trying to make, the main point, probably the main conceptual point that I'm making here is that when we decrease the flow rate from relatively low flow rate here, 64 nanoliters per minute, to 12 nanoliters per minute, we increase the signal to noise quite dramatically. Of course, it brings benefits and some also disadvantages or limitations, which I'll talk about in a little bit. But the main gains in signal to noise and sensitivity are defined by those reasons listed in the bullet points on the right. So, and in my lab, we are exploring mostly, and today I'll be talking mostly about porous layer open tubular columns, but we are using all modalities of separation columns shown here. Monolithic columns, which are polymeric sponges, polymerize inside the column, porous layer open tubular, which I'll show in more detail, and bead-packed columns, as well as capillary electrophoresis, and we are using sheathless electrospray. So the main differences between the bit-packed columns that we all are using, and the field is using bit-packed columns all the time, and the porous layer open tubular columns. So if we take a look at these TM images, and these are capillary columns, the same 360 micron OD outer diameter, the same polyimide coating. And the difference is in the inner diameter, where we are using 75 or 100 micron ID columns in the bit pack, and 5 or 10 micron ID down to 2 micron inner diameter in the plot columns. And as you see, if we zoom in into these perfectly spherical beads, some of those are not that perfect. And here we are generating a covalently attached porous layer attached to the inner wall of the capillary. So we need to operate those columns at substantially lower flow rate to benefit from the gains in their performance and to increase the sensitivity. And there are quite a few limitations currently in the field. The pumps delivering ultra low flow at about 10 nanoliters per minute or 20 nanoliters per minute are not that readily available. What we are doing, we are using regular pumps. We are operating them at about 300 nanoliters per minute or 500 nanoliters per minute, and we split the flow. To make the setup functional, yesterday Ryan discussed the benefits or potential disadvantages of using trapping columns. So possible losses in a flow through, and possible band spreading, so the separation performance can be compromised somewhat. However, to operate our ultra low flow columns, we cannot load sample at realistic sample volumes of 1 microliter, 2 microliter volume in a reasonable amount of time without using trapping columns. So to load our sample in the volumes I just mentioned, 1 microliter, 2 microliters, we have to use the trapping column, either bit-packed or monolithic columns. That's what we are exploring in the field in my lab. So we load them quickly, and it takes a minute or 2 minutes, depends on the sample volume. We can load up to 5 microliters, 1 microliter, whatever we want. And then we switch the configuration and we stop splitting the flow here, and we split the flow in the first T, and then we generate 20 nanoliters per minute going through the analytical plot column. So that's how we operate regular conventional non-LC systems with the split flow and the valves. So, and we are making our own tips. We pull them. We have tricks here and there. So, and that's the general setup that I'll be discussing pretty much in the rest of the talk. What is new here? We published 15 years ago pretty impressive results in MCP, low zeptamol detection limits. 5 zeptamols, actually we had results down to 1 zeptamol, didn't show it in the paper, but we looked at the cells even now for cells isolated from whole blood. The numbers are pretty impressive using the equipment that we had at that time. So, but unfortunately, these columns never picked up. So they were never used broadly used in many labs. And there are challenges. So not that easy to operate ultra low flow. So splitting is needed, then experience is needed. And also what we figured out, that you see those bumps, and if we zoom in, maybe on the next slide. So this structure, these little balls, they can be relatively humongous. We are using free radical base polymerization, and those structures, they can be detached from the column. They clog the tip, they clog the column. Sometimes we just polymerize the column and it's clogged already. It was not usable. So what we did with Michal during the last couple of years, we optimized the polymerization procedure. We optimized the morphology of the plot column. So now it's pretty smooth, very thin layer of the porous structure. And now the lifetime was expanded, the reproducibility was expanded, and usability was substantially improved. So these are the main advantages. So that's where we started. These ball-like structures, they were clogging the column. And actually, and that's the new morphology, new design of the porous layer that we are using right now. And actually, the thickness is shown here, about 150 nanometers. And what we found out, we polymerized, we used the original method to polymerize columns, and we just cut the column in several places. And we saw those structures again and again in multiple places. Now, when we do the same thing, we polymerize column, cut it in several sections, very reproducible morphology, very thin layer, no this bulky stuff inside the capillary. We do it next day, the following day, and so on. So it's the procedure is dead on, optimized, and that leads to the improved reproducibility and lifetime. So if you compare multiple runs, HeLa from Porous, injecting nanogram, sub-nanogram, a few nanograms, the runs look alike for different columns, different days, different runs. If you look at the reproducibility between multiple columns, multiple runs, either number of IDs, peak shape, peak width, so the reproducibility is there. And no matter on which day, no matter when we polymerize, so that's the major improvement. And then if we compare the performance of the plot column base setup with the trapping column up front, we use a short monolithic column here with bit packed columns. So either commercially available or packed in-house, so in the lab. And we go from low sub-nanogram amount up to 50 nanogram level amount, proteins on the left side, peptides on the right side. And we see about 50% more or so peptides when we use the plot column for exactly the same sample, HeLa digest. And when we get to the 50 nanogram level, the gains are leveling out. We are not getting any more gains because the capacity of those columns are relatively low. So we benefit more in sub 50 nanogram levels of sample load. So that's the message. And all these tests and benchmarking of columns was done on bottom-up samples, digested HeLa, standard from Porous. And then we are using the same.
What's your data? DDA,
in this case, DDA only, Lumos Fusion, 3-generation older instrument than you would use. But at least we had a good control, the same instrument, the same method, the same data acquisition. Now in timsTOF DDA, I didn't include the slides. We had 5,000, 6,000. So it has to be also benchmarked in a good way. Anyway, then if you use the same setup, we change the trapping column, and now we are looking at top-down intact proteins. We just sonicate, open the cells and spin down and inject aliquots of 200, equivalent to 200 cells. Good reproducibility, again, between the runs. In this case, we use FAMES. It doesn't matter. It can help. You may not use it. And I just have a couple of slides. Very nice peaks, sharp peaks for proteins, which is rare for intact proteins. Again, reproducible, covering quite a broad dynamic range for intact proteins, which is important. And then we are getting quite, for 200 cell equivalent, in one run, we are getting about 330 proteins, 7 to 800 proteoforms, which is, again, using older instrumentation, even comparing to the leaders in the field, quite impressive. There is definitely room for improvement. And then I looked for Michal earlier today. He's working on cell-in-one and injecting single cells to the columns, but this is an older slide. He injected 6 cells at some point. We got some coverage, mostly in this case, higher abundant proteins. And I will not go into details. Multiple isoforms, which are not like histones, isoforms, which are difficult to detect using bottom-up approaches. Now switching gears and a few slides on using the same concept of porous layer open tubular column, but as a trapping column in C. And we were quite excited by the results we published during the last couple of years, where we used CMS to show single cell glycomic analysis, actually surface glycomic analysis, and single cell top-down proteomics analysis. First papers in both fields and in both directions, and Yunfan and Annelies worked on that. Also, the same method was used to characterize blood isolates. So, but now we implemented the plot column and applied it, polymerized it in the inlet of the CE capillary electrophoresis. So about 5 centimeters or so, 5-7 we played with lengths. And we inject the sample, keep it there for less than a minute. It gets absorbed to the stationary phase. And then we elute in several runs. In the flow through, for example, we can look at metabolites, glycans, and then we can elute, let's say, for top-down protein intact proteins, or if you digest peptides, and then lipids. So the concept is to use that technology for multi-omic analysis of small samples. So, and the initial results, so the columns look a little bit different. This is a 30-micron internal diameter column. The morphology is somewhat similar, but different from the plot columns that I showed for LC. Yunfan polymerized them. Again, no bulky structures. They're not easy to clog. And initially, we tested the columns for bottom-up proteomics, HeLa, digest, eluting in several fractions. So the flow through here, replicates in different runs, then 10% acetonitrile, lower hydrophobicity peptides, then higher hydrophobicity eluted at 20% acetonitrile, 40%, and so on. Here, 4 fractions for each sample, and analyzed in 4 different runs. In the next iteration, we analyze everything in one run. So we inject the sample, it gets absorbed here, then the flow through is pushed a little bit from that plot section, separated by a plug of BGE. Then we elute with, let's say, 10% acetonitrile, again, a plug of background electrolyte, another elution with, let's say, 20 or 30% acetonitrile, and so on. So 4 fractions within the same run. And if we previously, and we published just CEMS, we had to stretch the separation to about 45 minutes to get more or less good data. Here, 1,500-1,600 proteins, intact proteins. But we had to slow down the separation to get those proteins detected. Now, when we combine those, in this case, 3 fractions, flow through, we combine with 5% elution, 11%, 40%. And these are replicate analysis of the same HeLa digest. Within the same run, about 16, 17, 18 minutes, we basically cut down the time of the analysis 3-fold, got here an increase of 25% in IDs. Fresh data, probably 2 weeks old or so. So that's fresh from the press, not from the press, but from the lab we are working in this direction right now. And then the idea here to move on, we kind of worked on different aspects. Look at top-down intact proteins, and we published on that. And actually, at the end, we typically see peaks corresponding to lipids. And a bunch of lipids are shown here. We didn't work yet on separating them better to increase the depth. And so I'll not go into more details. On the right side, in the flow-through and negative mode ESI, we are looking at glycans and metabolites. And so I just included one slide on that. We have quite a few initial data points there. So that's the concept, that's work in development. And with that, I just give you two choices. Either continue exploring bit-packed columns, or consider in the future plot columns as a red pill versus the blue pill. Thank you for your attention. Thank you to the funding institutions, and thank you to the lab and collaborators. Thanks a lot. That's the talk.
So with the multi-step elution, where you just have discrete concentrations of acetonitrile, can you comment on the quality of separation without using a gradient?
You mean on a CE part, right? You're talking about elution on a CE.
Oh, I was under the impression that this was also plot columns, the last portion that you presented, that was CE?
Yes, the last
Oh,
portion is all that's AC.
Different concentrations just to dislodge the material, and then you use electrophoresis to separate. Okay,
so that part is responsible for CE separation. High voltage is applied 20 kV, 25 kV across the column after we elute.
Okay, yeah, I understand. That's very helpful. So another quick question with the plot columns, you're getting the superior performance for very small samples. And do you have a sense of how much of the benefit is coming from improved separation versus the lower flow rate and improved ionization? Combination of both, perhaps, but what do you think is most important?
I think right now, mostly from ultra-low flow. Definitely a combination of things potentially, but as I mentioned, trap column may distort the separation a little bit. So if you do the same experiments without considering any time delays, the separation is much better. With the trap column, we are using a compromise, but we are working on practicality. Potentially, we need to introduce some refocusing, reshaping of the band, which we played before, but we need to do that. So if we have refocusing, we can continue to benefit from the improved performance as well as the ultra-low flow rate, which is translated into the better ionization efficiency, ion transfer, and utilization.
Okay, maybe another very quick question. So with the electrophoresis, where you have the plot column loading, is it something that you think you can automate? The sample loading?
Oh, it's automated. It's from autosampler. We just inject and then inject the BG from another vial, inject the elution buffer from another vial. It's all automated.
Cool, thank you.
Sorry for the confusion. I went through this fast. All right.
There are no other questions, then please join me in thanking.
Thank you.